All right, everyone else to find a seat. We get started here. All right. Tonight's guest speaker is Jordan Weeks. Jordan has worked in the Wisconsin DNR since 1994. In 1999, he received his Bachelor of Science degree from UW-Stevens Point, double majoring in both fisheries and biology. Jordan has worked at the Escanaba Lake Fisheries Research Station, as well as in Monaco, Woodruff, Eau Claire, Spooner, and La Crosse. In 2005, he earned his master's degree in fisheries from UWSP, researching comprehensive walleye and muskellunge movements in the Manitowish Channel Lakes. Jordan currently works as a leader for the Mississippi River Fish Team and the Muskie Standing Team for statewide muskie management. He's also Wisconsin's representatives on the ESOCID Technical Committee, part of the American Fisheries Society, working with fisheries representatives from throughout the greater Midwest and Ontario, Canada. In addition, Jordan has been Muskie Hunter magazine's research editor since 2006, authoring more than 80 articles and orating more than 100 public presentations. Jordan began fishing for muskellunge at age 16. He uses science in conjunction with muskie biology, life history, habitat use, diet trends, and past angling experience to help shorten the time between catches. He also enjoys teaching others how to use science to catch more fish. Jordan works closely with Lake X Lures, TNA Tackle, St. Croix Rods, Stealth Tackle, and Muskie Hunter magazine to make muskie catching easier. Everybody, please give a warm Titletown welcome to Jordan Weeks.
Thanks, Phil. I don't know who wrote that thing, but he's full of it. I just want to start out by saying thank you for having me. I've never been to this club before, and I've been to a lot of the muskie clubs in the state, but I'm really excited to be at this one. That I've heard nothing but great things about this club. In addition, I just wanted to thank you guys for all the help you give the department getting these muskies out here in the bay, because that reclamation project is really important to the department, and I think obviously to us as anglers. So, musky genetics, right? It's a very interesting topic. I'm going to start out by saying I am not a geneticist. So don't be afraid. I'm not going to go into loci and double helix and all that stuff because, first of all, I don't really know that stuff that well. But I can interpret some results from some of the research that's been done. So what I'd like to do tonight is give you a little bit of background about general muskie stuff in Wisconsin, then go into 6 case studies that show how the use of genetics can give us kind of a clue as to what's going on in Wisconsin lakes. They're mentioned here, some of which are published studies, some of which are thesis. If you're interested in reading more about any of these, I have cards up here. You can come talk to me later on. I can get some contact information and provide this stuff to you. Although if you ask to read both thesis, I'm going to think you're crazy. Then at the end, I want to talk a little bit about fishing. I got that little bee that the young man took. Somebody took it. Great. I have another one, and I have the other new lure from Lake X as well as the B52, and I have the new lure from TNA to show you guys at the end. So I always like to start out with giant, grandiose fish pictures. Now, you guys are no strangers to big fish because you got a whole bunch of them out here. These fish, I would say, are a little bit different in the fact that a lot of the fish I'm going to be showing you, in fact, the majority of the fish I'm going to be showing you are indeed stocked fish in Wisconsin. This right here, you can see this gentleman, that's a 300-pounder. This is a 50-pounder. This is a stock fish. This one happens to live in a lake in northwest Wisconsin. We've actually captured her a couple times. An absolute beast of a fish and just over 50 inches. So we do indeed have some large fish that have been stocked in the state. Is kind of the moral of the story there. Most of you probably know the distribution of muskies in Wisconsin. This map generally just shows the native range of the fish. Includes basically the northern third of the state and then also the major river drainages. When we talk about, within the department, when we talk about muskies, musky waters, we talk about in 2 terms, classification and reproduction. So when we talk about musky lakes class, we have 2 class A waters. A1 are trophy waters, A2 are action waters. A1 waters tend to be lower densities, the fish grow larger. These tend to be larger lakes. A2 waters tend to be higher densities, great lakes to take somebody to catch their first fish or catch a whole bunch of fish during the day. We have Class B waters, which may or may not be stocked on a regular basis. They are managed for muskies, but they're not super high on our priority list. And then there's Class C waters that are not necessarily managed for muskies, but they do have muskies. These Class C waters are your quote-unquote Lake X's that you find. They're typically connected by a stream, or they have muskies in them because someone used their live well, which I don't recommend. Although the fish tend to grow really large in these lakes because there's only a couple. Looking at it a little bit different way, hopefully you can see in the back, we have 123 Class A waters, or trophy waters, average size just over 2,000 acres. We have almost 200 action waters, over 200 Class B waters, and as you can see, going so forth. The tricky part is the rivers, the river portion. It says river segments for a reason. We don't have 100 rivers in Wisconsin that has muskies. We have segments. Our biologists are responsible for mostly for counties, and so when a river goes from one county to another, it becomes another biologist's responsibility, which is why we manage them in segments. Which is also why rivers are really hard to manage in Wisconsin. Not only that, rivers are really hard to sample from a fisheries perspective. We typically get our best results, which I'm sure most of you have seen out here on the river where they pike net muskies. That's the best way to catch muskies. We also use electrofishing. That's a really poor way to catch muskies for the most part. Mostly because their lateral line is very long and it's ultra sensitive, and they feel that electric current coming towards them and usually blast out of the field and we don't get them. Having said that, the largest muskie I've caught via electrofishing personally is a 40-pound fish out of the Wisconsin River in the Merrill to Brokaw section, but that was just pure luck. When you get the big fish with electrofishing gear, it's pure luck, or you're specifically targeting those fish. That technique is not commonly used in Wisconsin, but it's common. It's more common in Minnesota where you have these really clear lakes. What they typically do is they'll spotlight till they find the fish, drive over with the boat without the electricity on, and get over the fish and flip on the juice. And they can get them that way because they can't feel them coming. So we don't have as many clear waters as Minnesota does, so it doesn't work in all the places around here, but that's another way we can get them. Again, not as efficient as pike netting. When we talk about density of some of our waters, to kind of give you an overview, this is the average of all the lakes in the state, so it's really an average of a lot of waters. So our trophy waters tend to have about 1/4 of a fish per acre. Our action waters are about twice as dense. Gone are the days where we have a lake wingrow with 7 adults per acre. I don't know how many of you remember that, but it was dumb. Go down there and catch 8 or 9 fish in a day. We stocked too many fish in there at that point. Stocking is a real contentious issue right now because in the last 15 years we haven't got any license fee increase, and therefore our budgets haven't increased, but the cost of everything has increased. So we can't raise as many fish as we once did with the same amount of money. So like I said, gone are the days where we have fish stocked at 2 per acre every year and get a density of 7 fish per acre. It's just not going to happen. Class B waters kind of mimic our trophy waters, and that's why I put it in there, because most of us fish are Class A waters. Those are the most popular waters, those are the most well-known. But if you're looking for an out-of-the-way lake that has potential to grow really big fish, our Class B waters are where it's at. They have lower densities, very similar to our trophy waters. They tend to be a little smaller lake than our trophy waters, but they have the same potential. This column on this far side here, if you're not familiar with that metric, that's proportional stock density. That's the percentage of fish over 40 inches in that sample that we took. It's not total in the lake. It's not a model of, it's not an estimation based on the number we caught. It's the actual number. So if we caught 100 fish, 12 of those fish were over 40 inches. It doesn't tell you if they're over 50, it just says over 40. The other way we measure musky lakes in Wisconsin is reproductive category. Category 1 are all natural reproduction. Can I get a guess on what people think about how many lakes in Wisconsin have natural reproduction? Just a percentage. 5%? 20? Did I tell you that?
No.
You can't be that smart.
I'm that dumb just to guess it. Right?
Good job. You win a sticker. About 20% of our waters, and that might be on the high end. Musky reproduction is really hard to measure. as you probably all know, I'm going to say this a lot about Green Bay. I'm not going to talk specifically because I'm going to assume you're all experts on Green Bay. But it's really hard to find young muskies in a small inland lake in northern Wisconsin. Imagine trying to wrangle one out here. There's just so much water. The likelihood of grabbing them, even if they're reproducing at a really high rate, is the difference between getting a 0 and getting a 1 is negligible. Like, that doesn't mean anything. So finding young muskies and finding natural reproduction is really hard. And for a long time, the department has assumed that a lot of our lakes are NR lakes, when in reality, when we stop stocking some of these waters, the muskies go away. So category 2, we believe have some natural reproduction, but are supplementally stocked. And then category 3 waters don't have any NR, and we don't stock them. Category 4 waters are synonymous with our C classification. These are the lake X's. These are the connected waters that don't have direct stocking. The fish have fins, they swim into them. Low density can be big fish waters. These are the secret lakes that nobody wants to talk about. When we talk about muskies, ultimate length is the one we always talk about. Most of the inland lakes in Wisconsin, which is the data that I'm talking about here, can't compare with the ultimate length that these Great Lakes fish get out here. So we're going to put that to the side for right now. But when we look at all the information about ultimate length of muskies, which is this metric on the left side, that's L infinity. That's the average asymptotic length of the largest fish in the sample. We can see that 70% of the variation is explained by lake size alone. So what I'm trying to tell you here is big lakes make big fish. It's as simple as that. When we look at all the variables associated with muskies in Wisconsin and anywhere, lake size is the same thing. You think about it like this. You go to the carnival and your kid wins a goldfish. It's in this little bowl, right? You can feed that goldfish and feed that goldfish and feed that goldfish. It's only going to get so big. You take that goldfish and you put it in a 100-gallon aquarium, it's going to get big. You put it in a 1,000-gallon aquarium, it's going to become a carp. So it can be explained by lake size. The key trigger that we found with our data in Wisconsin is that a lake needs to be, on average, 2,000 acres or larger to produce any measurable number of 50-inch plus fish. We don't have a lot of lakes like that. Wisconsin has a lot of small lakes. Average lake size is about 800 acres. That's our average muskie lake size. That's all of the lakes put together, all the classes. And so there are actually only about 25% of our Class A1 lakes that fit this category. Lake size is a really big indicator. If you want big fish, you go fish big water. So last year, were any of you at the Milwaukee Expo and caught my seminar there? Well, great. This is new. I should have just done that one.
That's why we brought you.
I didn't have to do any extra work. Okay, so last year, I was sitting around thinking and talking to some of my colleagues and kind of hypothesizing what would be a good topic for that seminar. And basically what I came up with was I wanted to compare catch data from Wisconsin versus other places like Minnesota, Lake of the Woods, and the famed Leech Lake, right? So I went to the Muskies Inc. database. By the way, I am not a Muskies Inc. member, so I have never registered any fish in here. But I assume, well, let me back up. If we assume that all of you guys lie the same, then this is really good data, right? So that's why I put myself aside, because I lie better than you guys. So anyway, I mined all this data. I went in with the Wisconsin, the data from Wisconsin from 1990 to 2018 crashed my Excel spreadsheet twice. There are so many entries, it's unbelievable. Wisconsin blows everybody else out of the water. It's incredible. Anyway, so what I did is I looked at fish that were 45 inches and larger, 48 inches and larger, and 50 inches and larger, and I apologize for the guys in the back. You're not going to be able to see this, but I'll do my best to kind of put my hand where the levels are. The black bars, the ones that are the highest, are 45-inch fish. You can see a noticeable increasing trend through time. 1990 over here, 2018 over here. There's a blue bar that goes up like this. Those are 4-footers and larger. Or excuse me, yeah, 4-footers and larger. And then there's green bars that are here in a smaller level, and they also increase All these are statistically significant increases through time of fish that are 45 to 48 inches, 48 to 50 inches, and 50 inches and larger in Wisconsin. What I'm trying to tell you with this,
Did you include Green Bay?
I did not.
Okay.
It was all inland waters,
That's what I was just curious.
I think. Good question. I'm going to say inland waters. Can't remember. It was a year ago. Come on. The important part here is we're catching more big fish than we have ever. There's a number of reasons for this, right? I don't have data to prove what I'm about to tell you, but I think that most of you will probably agree with this synopsis. So I put 3 red arrows on here. This first one over here, I'm going to call the beginning of catch and release. That's about 1996, 1997. One of my good friends, Steve Heiding, was guiding at the time. He told me, I was asking questions, he told me that in 1996, that was the last year any of his clients asked to keep a muskie. So I don't know, I'm making that up, that's the date, but it's pretty close to when it was widely accepted by everybody, we're not going to kill these things anymore. The second arrow is also kind of a general timeline. That's around 2006, 2007. Talked to Jim Sarek, he said that was 2007 was the first year his show was on TV. My estimation, I'm going to call that the mainstream muskie media blitz, right? There was, message boards and crap before that, but it was like a small community generally. It went mainstream when these TV shows went on. I'm not sure when Key's show started, probably somewhere around there, maybe a year or two before. I didn't ask Mike to find out. But anyway, that is pretty significant. But what you can see is muskie catches were going along pretty level here as we were killing them. Right? When we stopped killing them, you can see a precipitous increase in the number of fish, the number of big fish that survive to be caught by anglers. You can also see an increase after that media blitz. I think that's because the fishing for the species got popular, right? More people decided to do it. Theoretically, that means more fish get caught. When more fish get caught, theoretically, more big fish get caught. The last arrow over here on the far left is one I put in there because I don't have any data again, but I feel really strongly that this was a huge aspect of muskie conservation in Wisconsin in particular. That signifies the prohibition of single hook sucker rigs. Does everybody know why I'm saying that? If you don't, I'll tell you. For years and years, Wisconsin had a very long history of sucker fishing, and the most common way to catch them at the time and back in the day was to put a big square number 10 hook in the nose of a sucker. When you get a pickup, smoke a cigarette, drink a beer, and then you reel the fish in. The fish subsequently has swallowed the sucker, and you get it in the net, take a picture, clip off the wire leader, let the fish swim away. They all swim away, every one of them. What we didn't know at the time, I'm not blaming anybody, nobody knew, 80% of those fish are dead within 6 months. That hook does not dissolve. It typically rips the esophagus or stomach lining, causes infection, and the fish dies. The reason we know that is because one of our longtime researchers and muskie guru, Terry Marganeau, had suspicions about this happening. He grabbed a bunch of adult muskies from the Spooner area, put them in the hatchery, caught them with various rigs, and watched them die. So this isn't really something that we can debate. 80% of these fish are done, which is why we made it illegal, and which is also why if you ever see anybody using one, you should call the game warden. So we haven't seen a precipitous increase yet, but we have to remember that muskies are a long-lived fish, and I firmly believe we are going to continue to see this upward trend of large fish being caught in the state of Wisconsin. I personally have caught more large fish in the last 10 years than I did before. I'd like to think it's because I'm better, but it's probably just because there are more big fish out there. All right, on to the topic at hand, genetics. Genetics. Does anybody remember like 2002, 3, 4, those years? I can barely remember them. But what I do remember about those years is there was this stock of fish that people were catching that were growing really large and were believed to grow like superfish. They were from a lake in Minnesota. I think it starts with an L. Leech, that was it. Yeah, so there was fish in Minnesota being caught at a really high rate, and they were really large. They're really stupid, and they were caught in high numbers. That spurred a whole bunch of debate within the musky fishing community in Wisconsin and therefore made the department look really closely at what we were doing as far as stocking fish in the state. So when we talk about genetics, it's one of those things where, you know, we have beliefs, or we had some thoughts about the process, and all of you may have thoughts about that, about what you think is going on, but I'm not necessarily here to tell you what to think. I just want to ask if you're willing to. Because what I'm about to tell you is probably going to go contrary to what you may believe. And I realize that that's a hard thing to kind of deal with. And again, I'm not trying to tell anybody that they're wrong. I just want to provide you with some data. And if you choose to believe it, then great. If you don't, well, that's okay too. But before we do that, I need to go through some terms so that I'm not speaking in some foreign language to you guys. I tried to make this as simple as possible, and I'm not trying to insult anyone's intelligence, so if you know all this stuff already, I apologize. But it's something that I find that helps a lot of groups understand the musky genetics portion. So when I talk about a stock, most musky anglers use the word strain as the same thing. In the scientific community, we call those fish stocks. I'm not exactly sure why that is, but all the geneticists that I talk to get kind of upset when you start saying strain instead of stock. So I just want to let you know it's not wrong to call it the Leech Lake strain or the Great Lakes strain. It's just if you're a geneticist, they get a little squirrely about that. Genotype is the actual makeup of the DNA. That's the DNA strands that make us all different. Phenotype is how the fish looks. And for a long time, the way a fish looks was the way you would delineate which stock it was, right? But as we can look around the room, we can see that we're not all from the same stock, right? Some of us are tall and thin, some of us are short and thick, some of us are very short and thin. That's our phenotype. That's what we look like. The muskies in the natural world all can look a little bit different. They can all look a little bit different in various waters, especially when they're together. Talk about fitness a little bit. That's the ability to survive and reproduce. The definition of reproduction is when a muskie successfully spawns, that egg goes to the bottom, hatches, that young muskie grows up to drop another egg or milt. So the definition of reproduction is you actually have to replace yourself. And a lot of people use that in the incorrect term, right? We say, is there natural reproduction? Well, because I saw two muskies, they were paired up swimming along, they reproduce out here. You know, that's like practicing in college. Sorry, young man, you'll learn someday. It's the same thing, right? This is the tricky one, this outbreeding depression or introgression. It's not the same thing, but they're very similar. So it's a reduction in that fitness due to crossing of various stocks, right? It may not happen in the first generation, but as we see more and more crosses, these fish tend to have less fitness. They have lower survival, lower growth potential, and lower reproductive capabilities. Progeny is just offspring, so sometimes I use that word and people start their eyes roll back in their head. I try not to do that. Here's a couple other stock fish just to keep me away from the data. I saw a few of you falling asleep. These are extremely large fish. The one on the left is from Pewaukee Lake, and I think this super tanker is from Okauchee, I want to say. 55-gallon barrel there. That one's built like me. So to get into the actual genetic study, and it's awesome because no two genetic studies are going to find the same thing, and no two waters necessarily are going to show you the same thing. It's really, really cool stuff to look at. If I could go back and do it again, which I guess I could, but I'm too far down this rabbit hole to turn around. I'd be a geneticist because this stuff's super cool. So this is Lauren Miller's study. It was published in 2009. I actually wrote a Musky Hunter article about it. Don't ask me when because I have no idea. I write an article and I forget what I wrote and where I wrote it, and there's been so many of them. But they did a research project on Moose Lake in northern Minnesota. And if you don't know the story about Minnesota muskies, there was Leech Lake, which had that stock of fish that is what Minnesota muskies is now. But that was separate from what the state propagation program was doing. They used Shupak Lake, which is a landlocked lake in Voyageurs National Park in far northern Minnesota, as their broodstock lake for like 20, 25 years, from like 1960 to 1980. They used that lake because when they went in there, the fish were naturally reproducing like crazy. There was a whole bunch of them, and they could get all the eggs they wanted in a really short amount of time. When you work in a hatchery, that's like the best, because it's easy. right? You go there, you throw out a couple nets, males and females everywhere, gametes all over the place. You can raise as many as you want, really easy. What they didn't know at the time is those fish generally don't get over 40 inches, ever, anywhere. I don't care if you put them in Mille Lacs, you're not going to get much more than about a 42 incher. And that's fantastic. So they didn't know this at the time. So in the early 80s, late 70s, the Minnesota Department of Natural Resources was getting a lot of complaints from their anglers. They're like, hey, we're catching all kinds of muskies. These fish seem to be reproducing. There's a whole bunch of them. We can't catch any big fish. So they started looking at this problem. They said, okay, they identified that, okay, these lakes that we've been stocking a long time with the shoepack fish tend to have small fish. And Leech Lake doesn't. There's something different here. But at the time, they didn't have the genetic capabilities to assess these populations correctly. There wasn't the technology available to correctly identify these stocks, right? It was only in the early 2000s that we got the technology to be able to correctly identify these various stocks. Otherwise, they're just like, oh, this lake has that thing. This lake has that thing. And we assumed, based on phenotype, that they were different or the same. So Lauren Miller looked at fish in Moose Lake in 3 different time periods, which I'm not sure yet. 81, 86, 1990, and 2004. Moose Lake was stocked one time in 1985 with Wisconsin source fish. And I need to back up a little bit, because once Minnesota figured out those shoepack fish were garbage, they decided that we have to do something in the interim. They didn't have the infrastructure ready, and they couldn't catch the fish in Leech Lake to spawn them. They spawned a tad bit deeper, and they weren't netting the right area. So they asked us for some fish, being a good neighbor. We gave them fish for about 3 years, and they stocked them into a bunch of different lakes. Moose Lake was one of them. Once these fish were stocked into the lake, that was the last time Moose Lake got stocked. Because in doing this research, they found out that there was natural reproduction occurring in Moose Lake. So they said, well, we don't need to stock it anymore, which is similar to what Wisconsin would do. So they didn't stock after the Wisconsin fish. They realized it was naturally reproducing. And just for information, the density was basically what we'd call a Class A1 lake. Relatively low density and had some decent fish in it. So what did he find? When Lauren did the research, he found that there were 3 genetically distinct populations. Shoepack, there was a Moose Lake native fish that was always there, and there was a Wisconsin fish. In the 1981 sample, they only found 2. Why? Because Wisconsin fish hadn't been stocked yet. They found that the fish they've been stocking survived very well and provided a very large sample, 81%. The Moose Lake native population was hovering around 20% of all the muskies they caught. In 1986, they did the same genetic analysis. Basically, and what they did is we have scale samples, and we go do these research projects. Historically, we would take muskie scales to try to age them. Scales are a really bad way to age muskies, turns out, but we just found that out not too long ago. But we've had these catalogs. We have file cabinets in northern Wisconsin of scale envelopes full of these scales. All of those are great genetic material. They're really bad for aging, which is what they were collected for. So anyway, Minnesota had all this information. Looked at the 1986 sample. They found, again, a real high percentage of the stock shoepack fish. Again, they survive great. They reproduce great. 22% Moose Lake genetic, and we did see this 1-year-old cohort of Wisconsin fish showing up in the sample. So what happens next is the really important part, right? In 1990, after 5 years of stocking, most of the Wisconsin fish in Moose Lake at that time would be mature. About 90% of the males in Wisconsin mature by age 5, and about, this is ballpark, 50 to 70% of the females will mature at age 5. So the way we get these samples is when they mature, we catch them when they want to spawn, right? So that's how I know that they were mature. We did find a quarter of the fish then had survived from that Wisconsin stocking and made up the sample. But at this time, which is interesting, they didn't see any crossing. The important part is the next part, where they saw every single cross plus every single ancestry. So what we had is an example of that outbreeding depression or introgression in Moose Lake. Now, as a geneticist, they're like, this is not good. But what we hope happens in these situations is that through the reduction of fitness of those crossed individuals, through the reduction in reproduction, and through the reduction of survival, those fish eventually will leave the population. So the hope is in Moose Lake that eventually it'll get back to these guys. Unfortunately, the Wisconsin fish seem to persist really well in Minnesota waters, so they may not get lucky enough to get rid of some of those green fish over there. Really cool study, really enlightening information when it talks about what happens, right?
So are any of those crossbreeds stronger, or are they all weaker? Or
The-
no,
they
do they
t-
get bigger? Or?
Typically not. That's the reduction in, well, I didn't mention growth, but reduction in growth is typical of introgressed individuals or outbreeding depression. Typically, they don't get as large. And like I mentioned before, it's not so much the first time they cross, so the first individual, but when a crossed individual reproduces with another crossed individual, that perpetuates, like you procreating with your sister too many times. Which might just be once. I don't know. Do you have a sister? I don't even know her. I'm sure she's like just once.
I'm not sure about our kids.
Or your brother, whatever. You guys are probably familiar with this gentleman. He's one of my really good friends, Dr. Kevin Kapusinski. He was the biologist here for the department for a couple years. 2005-ish, maybe? Did I blow up your cameras?
Nope.
These are the ones that make you look thin, right?
Yeah.
Awesome. Perfect. Those are the best cameras ever. Anyway, Dr. Kevin Kapusinski now works up at Lake State in Sault Ste. Marie as a fisheries professor up there. This research he did in conjunction with UWSP Brian Sloss and Dr. John Farrell from Syracuse University. This was part of Kevin's doctorate dissertation. What they looked at was Great Lakes populations of muskies, and they contended that they would be genetically different from one another based on the vast expanses in which they occurred and the distance between populations. These were all the populations. Sorry, it sucks. That's a very terrible list. Essentially, Green Bay, Georgian Bay, St. Lawrence River, and a couple places in Quebec and Lake Erie. So it was spread throughout the upper Great Lakes. What they found was that there was significant spatial pairing. For example, in Georgian Bay, if you're not aware, on the north side of Georgian Bay, there's a number of different rivers that come into Georgian Bay. Each individual river has its own stock of muskies. They're all genetically distinct. In the summertime, they go out in Georgian Bay and eat fish all summer. You could catch a fish out there, take a genetic sample, and the geneticist would be able to assign it back to the river that they spawn in. They're all distinct. The Green Bay fish are a little bit trickier. We kind of have a hodgepodge. It started out with the Indian spread chain from Michigan. We got some fish from Georgian Bay. We now got some fish from Lake St. Clair. So what we're trying to do there is build up that genetic diversity of the Great Lakes stock, even though they could be separate. Not as worried about introgression or outbreeding depression because we don't really have a choice. We're basically taking fish from where we can get them, and hopefully our agreement with Michigan holds. They're a little bit mad at us right now, so we're hoping we can still get some Lake St. Clair fish. The good news regarding that, and maybe you're all aware, is we can now use Fox River fish. We have the ability to disinfect the eggs and take them into our hatchery, so we can use fish from the Fox to help raise more Great Lakes fish. So that's really good news for your club in particular. It's also very good news for all the other waters that drain into the Great Lakes, because Wisconsin's current genetic management plan is to stock all waters, or nearly all the waters that drain into the Great Lakes, with Great Lakes stock.
Don't we currently have fish though for wild rose? One more year?
Yes.
Okay.
Yeah, we got yearlings coming this year, for sure. And we're hoping that Michigan will give us fish from St. Clair again. They're just mad at us because we're stocking too many salmon. It's not even about muskies. Yeah, and that's not, it's a political thing. It's not within our control. It's not within the DNR's control. So, the other cool part about this is they actually saw that fish pass Niagara Falls. They survived going over Niagara Falls.
Wow.
Right?
Yeah.
So there isn't a dam in Wisconsin that's going to stop these things. I can tell you that right now. That's super cool. The populations that they found were similar. Were stock population. This Buffalo Harbor, Upper and Lower Niagara, are stock now. They're from Lake Chautauqua. And the reason they have to stock them is because muskies can't spawn on seawall. basically in that harbor area where they used to spawn, they made it, you know, industrialized, and it's all seawall, kind of like the river here. They can't do it there, so they've had to supplementally stock that area. Obviously, Green Bay is different, and then there was a couple other populations that were slightly different from the other ones. Really, another cool experiment showing that, hey, we have some significant genetic units, especially in Georgian Bay, that are relatively close geographically, but genetically different. These are fish from Rice Lake. That's a stocked water. That was actually started, I think, in the early 2000s was the first time that thing got stocked. Super productive water. If you've ever fished it, it may be worth a try. The third one I want to talk about is Matt Diana, working with the Illinois Natural History Survey. If that Diana name is familiar to anybody, his dad is Jim Diana, longtime University of Michigan muskie researcher. He's vying for the Mount Rushmore of muskie scientists position right now. Jim's a great guy, super knowledgeable, done a ton of publications regarding muskies. They had a hypothesis, and they did want to do some research in Illinois about survival of muskies. This, again, had to do with the popularity of the leech lake fish and people wanting to stock those things all over the place. These guys hypothesized that, well, while we may be able to stock those here because all our fish are stocked, we think that the fish that are here already are best suited to live here. That was their idea, and they wanted to test that. So they got a bunch of fish from Leech Lake. They also had a stock from the Ohio River, which is the same fish that are in Chautauqua, Clear Fork, and Cave Run. And then they have this really interesting Illinois stock, they call it. And it's a cross between Ohio drainage fish and Mississippi or Leech Lake fish that they've had since 1993, which seems like a bad idea, knowing what I know about introgression. And if you are familiar with what's happening in Lake Shelbyville at the moment, it may have reared its ugly head. In the last couple of years, they've had significant muskie kills there, where they've had a whole bunch of fish die, and it could be because of some resistance. It could be a disease, could be something else, or it could just be the lack of survival because they're so introgressed.
Didn't the lake just produce the record fish though, just a few years ago?
If most of the other ones died, it maybe did, because that would have a lot of room, right? I don't know. I'm not saying they all died. I'm just saying they had a large kill recently. I don't know that answer.
That's where the Illinois state record's out of, isn't it?
I still don't know that answer. I mean, I know the bears suck. That's all.
Yeah.
So they had 3 study lakes: Pierce, Mingo, and Samdale. Pierce is near the Wisconsin border. Samdale is sort of in the middle of the state, and I lied. Mingo's in the middle. Samdale's in the south. They stock these various stocks of muskies in the lakes, and they followed their growth. What they found was that within the first 48 hours after stocking, that they had some significant mortality. 28% of the Ohio fish died, 8% of the Illinois, and actually a really small percentage of the Mississippi River fish died. This was in the first 48. They kept them in a holding pen. What they found out was the kicker, and probably some of you know from stocking fish on the bay, is the higher the water temperature, the worse it is for muskies when you stock them. It's a couple reasons. Most of the time, the hatchery system waters are typically cooler. When you thermally stress a fish by changing their temperature too much, that can be really bad for them. The other thing about stocking muskies into warm water is that species like bass are really active, and there's nothing an 18-inch largemouth wants to do more than eat a hot dog, and they can really put a hurting on muskies. I used to work in Vilas County, and we'd see it all the time. If the water was too warm, pull up to the landing, and the bass would make the water boil, eating those things. So the moral of the story is you want to stock muskies at the lowest water temperature you can get. 50 is kind of the magic, like, get out of jail free number. Bass get really lethargic at 50, and muskies love 50. So stocking muskies at 50 degrees is actually the ideal temperature. And the department is moving I forgot to mention I'm a leader of the muskie team in the state, and we're moving towards trying to get our hatcheries to stock a little bit later in the year, which does two things. It allows the fish that we grow to get a little bit bigger, which is always a good thing, and it also allows for better survival, having those water temperatures get a little bit lower. So what they also found that they caught them at, excuse me, let me back up. When they went out to survey these fish, they caught them all kind of at a very low rate, but they caught them all in proportion to where they were, the densities that they stocked them at. Over the first winter, they all survived pretty well. 40% of the fish that they stocked survived. If you told me that was going to happen, I'd be jumping up and down. 40% of the fish, if we told you 40% of the fish we stock in the bay are all going to live, like that would be, woo! That'd be really great. So they had good survival. The following summer, they found that the Illinois fish survived the best, followed by Ohio and the Mississippi River fish. And then after the second year, their first full year in the lake, survival didn't differ. So they found that survival of all the stocks was not changing through time. When we looked at fish length, after the first winter, the Illinois and Ohio fish were larger than the Mississippi River fish. But then after the second fall, so they're 1-year-old fish, they were all about the same size. So the Mississippi River fish caught up. Second spring, again, they found that the Illinois and Ohio fish were larger. Now this could be because those fish maybe actually grow in the wintertime, whereas the Mississippi River fish weren't. And that can be possible. But the results that they indicated in this paper, obviously, like I mentioned before, stock muskies in the coldest water possible. And they found that survival of the local stock was the best overall. So those weird Illinois fish that they've been raising since 1993 survived the best. They survived better than the Ohio fish and the Leech Lake fish. Mississippi River fish were consistently smaller by the end of the study. Hypothetically, they said because they didn't perform as well. It really kind of makes sense when you look at this stuff. We've seen it a little bit here in Wisconsin too. Locally adapted fish perform the best. Leech Lake fish do the best in Leech Lake. Wisconsin do the best in Wisconsin. Here's some. These are not stock fish, but these fish are significant enough to show. The smallest fish is on the far left. That one was 52. This one was closer to double nickels. And those are northern Wisconsin and our fish. Never remember where they were, though. I bet you if you found this guy, you'd find out. What?
I said, are you sure you can't remember?
We'll see what you got in there, eh? What you got in there, Bob?
I'll throw it in the kitty.
The fourth study I want to talk about is not a published study as of yet. It's in publication. They're going to talk about some of this stuff. I don't know if all of you are familiar, but Lake Wissota is a large impoundment of the Chippewa River over by Eau Claire. It's been stocked with Leech Lake fish since about 2005. The first Wisconsin chapter over there asked to stock those fish in that lake, and they could do it because at the time it was deemed a universal receptor. There was a fisheries biologist that worked there for 30 years, and he delineated it as a universal receptor water because he could never measure any natural reproduction in the system. This lake is my home water. I mean, I didn't grow up there, but I grew up fishing there. My grandparents lived really close to there, and as a youth, I can remember catching juvenile muskies in the middle of the summertime. And so I tried to tell him that there was NR out there. He wouldn't listen to me, but I don't blame him. I mean, I'd have a hard time, just taking the word of some punk kid, too. So, anyways, it was deemed a universal receptor. First, Wisconsin got to stock Leech Lake fish in there. 05, 07, 08, 9, 10, 11, 13, and 15. So quite a few times. They ranged from 300 fish to just about 2,500 fish. And over that time period, 30% of the muskies stocked in Lake Wissota were of Leech Lake origin. The other 70% were Wisconsin origin fish, upper Chippewa fish. In 2015 and 16, we were doing a comprehensive survey out there. So all the muskies we captured, we took a fin clip sample for genetic analysis. The fin clip samples we take are about the size of your thumbnail. We put them in alcohol, send them to Point, and they can classify them for us. I actually was part of this survey. It was a whole lot of fun. We caught 156 muskies in those 2 years. Sent them over to UWSP. Only 4 of those fish were pure Leech Lake fish. I think that club club spent $120,000 on those fish. If that's my money, it's not a good ROI. Just saying. But those fish were of multiple year classes. Incidentally, we took pictures during the survey of anything that phenotypically looked different. We were trying to get at that, hey, it looks like game. Only one of the fish looked like it and was. This one was like a flat white fish. It was really weird. But that was the 40-incher. These two fish were aged via scale. I will caution you, scale ages aren't good. Could be off by 5 years there. But the folks in Eau Claire aged them at 10, a 37, and a 35-inch males. I don't know if I'd buy too much into that, other than there were 2 males out of the sample. This one, obviously, you couldn't tell if it was a male or female. The largest fish in the sample was a 50-inch upper chip fish. They aged that at 13. Again, you could add 5 years to it pretty easy. I wouldn't put too much credence in the age of that fish. It could be 25. The important part of this whole research that they found 4 fish that were crosses. So, despite the fact that we only caught 4, there has to be enough of them at least to reproduce in there with the upper chip fish and to do it multiple times, right? Because you have 1-year class of fish that are 12 and 11. That's 1-year-old fish. There's a 2-year-old fish here, which is 15 inches or so. And then there's probably a 4 or 5-year-old fish. So these leech lake fish were spawning with the Wisconsin fish at least 3 different times, 3 different successful spawning events. So this is probably a minimum, but I don't think it's a lot higher than that. And all really it would take is 1 fish, right? So this was an example of when fish got stocked into a system, they either didn't survive or they didn't stay there. If you know anything about Lake Wissota, it's part of the Chippewa River, like I mentioned. The dam is very open, especially during high water conditions. They could certainly have left and tried to swim back home. It's possible. My guess is that they probably didn't survive because I got to see some of these fish, and they were not impressive coming off the truck. When we ask our hatcheries to produce, on average, 11-inch fish, that's the minimum that we expect. And I know for a fact a lot of those fish were 9, which is not something that's probably going to survive very well in a system. So, kind of cool results. Ancillary to this, they've taken a whole bunch more samples. What happened there? Samples! They took a bunch more samples. We're having those analyzed right now, and the results should be back pretty quick. And again, like, it's going to go into a publication that geneticist at Point's going to do. So these admixed of individuals were particularly important because it demonstrated natural reproduction in the system, which is where I started out in this story, and therefore it kicked that lake out of the universal receptor list. So even if the club wants to stock leech lake fish anymore, our genetic management plan prohibits them from doing that. And my thought on the process personally is that, look, the Chippewa River was a stronghold of our native muskies forever. There's no reason we should put anything else in there other than Chippewa River fish. Now, if these fish are going to be stocked somewhere, they should be stocked somewhere where A, they can't get away, and B, they're not native musky waters. I don't care then, and nobody really should care, especially if you guys won't pay for them. But the other problem is, and the other aspect about the leech lake fish is, a lot of the anglers said, well, why don't you guys just raise them? We can't get them. In order for Wisconsin to get leech lake progeny or leech lake eggs, we'd have to ask Minnesota to kill 60 of their adults to test them for VHS. And that's not a DNR rule, that's a DATCAP rule. And if you guys can get Minnesota convinced to do that, come talk to me. Because I think I know what they're going to tell you. In fact, I do know what they're going to tell you, because they told me already. So, again, demonstrating this introgression hybridization in Lake Wissota, be it at a small rate, but it's happening. Potential loss of all the good qualities you want to have in muskies. All right, this actually was a fish from Lake Wissota, 52-inch Wisconsin fish that I caught outside of the sample period in a fight net. So that was a pretty large fish. She had 3 boyfriends, which was pretty fun. They were all big too. That fish, I'm going to go back, that fish actually it was caught. They used to tag fish in that club with floy tags, a little tag that sticks out of the back. It was caught by one of the club members, I think 2 times previous to that. And miraculously, that information wasn't given to the department until I caught it with the tag in it. I said, where's this information? It was conveniently left out. Interesting. These are the theses that I'm going to talk about. There are 2 really important theses that were done at UWSP, all surrounding this idea of, hey, there's something with the genetic in Wisconsin that makes them not grow to the same size as a leech lake fish, right? That was the premise. All these giant fish are being caught in Minnesota. Anglers said, why can't we do that here? Your fish suck. We're like, okay, let's find out. And that's really how some of this research started. It took a little while because, as in all government, nothing moves fast. We got some grad students on this with the genetics lab in Stevens Point, with the idea that we know muskies have been propagated in Wisconsin for a really long time. And these angler concerns existed regarding the genetic integrity of Wisconsin muskies. Some of you may have heard the term mutt. I like to use that often with these. That's a beautiful mutt in my mind, sort of mocking. The idea of mutt infers that these are genetically inferior fish, right? They're all the same because we've stocked muskies all over the northern part of Wisconsin, right? That was the idea behind this. We want to say, well, let's find out if that's the case. The objectives were to evaluate the variation of one of our premier musky waters, which was the Coode Ray. The Coode Ray was used as a brood source for many, many years for the Governor Thompson State Hatchery. The other lake that was used primarily was Bone Lake, and Bone Lake was actually created with fish from the Coode Ray. So Bone Lake fish were Coode Ray fish moved over to Bone. Okay, those were the 2 major sources of genetics for the Governor Thompson Hatchery for many years. And again, this was the time where the hatcheries were going to these lakes and saying, hey, we can get a lot of fish really fast and a lot of eggs. So they would just take the first few fish they got, take all the eggs they could, and that's how we did things. This was before we had genetic information. This was before we had the technology to be able to say, well, wait, there are better ways to do this. Second part of this research was to determine if there was genetic population structuring. Or, in other words, are all the populations the same, or are they distinct from one another, like the fish in Georgia Bay? So that's the 2 things that they wanted to do in this thesis. The Coode Ray was important because it was the traditional brood source for muskies in Wisconsin, beginning in 1939. That's a long dang time ago. It had a reputation as a trophy fishery. Whether or not you agree with or believe any of those old giant muskie stories from Cal Johnson and Louis Spray, some of those fish allegedly came from the Coode Ray. And in fact, a biologist that worked there for a really long time has documented 56-inch fish from the Coode Ray. So it indeed did, at one time at least, have some really large fish. So it's important. We thought, hey, let's look and see what happened there. We do know that that one has particular interest because it had been stocked at a couple other times with fish from lakes that most of the angling public would call not premier water. So the theory or the potential for introgression was there, which also made this a good category. Those were big spider and mud Callahan. I see you looking. I'll read for you. Both, again, were perceived to be slow growth, small size populations. And so did these suspect stockings change the genetic of the Coode Ray? So what we had in historical scale samples were samples from 1956, 66, 76, 1990, and 2006 from the Coode Ray. We had a sample from 1956 in Spider, 1979 in Callahan, and the first step in this process was to see if Coode Ray 56 and Spider 56 were different. And what they found was indeed they are genetically distinct. So they're separate populations. So if those fish went to the Coode Ray, spawned with the Coode Ray fish, we would be able to tell the difference. You see the asterisk here on 76? This 76 sample didn't assign with the Coode Ray, but it didn't assign with these other 2 either. When they did this project initially, it was like, what the heck's going on here? Completely different than all of those. and I'll get back to that in a second. Second step was to evaluate if Leucouterea 1966 and Mud Callahan 79 were distinctly different, and indeed they are. So not only is Mud Callahan different from Leucouterea, so is Spider, right? We got 3 different populations. We can tell the difference between the fish. Third was to test if Leucouterea had the same genetic from 1950 to the present day, or is it, for lack of a better term, bastardized by these other stocking events. Back to that 1976 sample. When they first presented this information to the DNR Muskie team, one of the biologists sat there and he looked and he said, "I'm gonna have to get back to you on that." It was Steve Gilbert out of Vilas County. He went back into his stocking records. He's got these file folders, these paper stocking records. He found where the hatchery in the Ardemke hatchery in Woodruff stocked fish in there in 1976. So we found where those fish came from. They're from the Woodruff hatchery, from the Monaco chain. So, using genetics, and then we matched it up with the Monaco chain over there, we're like, okay, now we know where that is. So now it actually gave us another sample to say, okay, did that have an influence? So, we looked at that. This is the kicker right here. None of those stockings changed the genetic makeup of Leucouterea at all. We can't find any evidence. Now, there's a couple things going on there. First of all, historically, Leucouterea was a naturally reproducing population. If you know anything about the lake right now, it is indeed not. The reason for that is a giant cranberry bog that was formed on Musky Bay, where all the spawning area was. The muskies don't spawn there anymore. Well, they spawn there. They don't reproduce there anymore. So, that could actually have been a blessing in disguise to maintain our Leucouterea genetic in that system. It didn't allow the mixing of those other stocks with the Leucouterea fish over time. But good news is the fish in Leucouterea are the same genetic makeup as those giant fish that Cal Johnson was catching with back in the day. So that's really good news, right? The second one was to determine what the genetic makeup is of northern Wisconsin muskies. And in particular, what they were looking at was naturally reproducing populations. Like we mentioned before, there's generally a very small number of NR lakes out there. So what they did is they specifically looked at 24 waters that had no stocking ever, super limited sparse stocking, or haven't been stocked in 10 years. It was really hard to find 24 lakes that fit that category. And when they did the analysis, they found that only the NR lakes were the ones that they were able to use, because they separated out very nicely. What this shows, and again, I apologize to the folks in the back, this is probably the best way to graphically show the results of a genetic analysis of multiple populations. And what this is it's called unrooted tree diagram, showing lake names on the ends of the lines connected by various lines. Now I put that red and black box around there. Where are these lakes? Who knows these lakes? They're in the east, right? They're in the northeast part of the state. Where are all these? They're in the northwest part of the state, right? So if we contend that our hatcheries have an impact on the fish populations throughout Wisconsin, this pretty clearly shows that it probably does, right? The other part of that is we do have 2 major drainages that happen to lie within those same areas, right? So it could be that. It could be the upper Wisconsin fish and the upper chip fish showing themselves. The cool part about this diagram is the length of the line and the degree in which they're separated actually dictates how far apart these populations are. So if you had a really short line next to another short line or coming off the same branch, that would mean those populations are very close to each other genetically. We don't see that here. We see a pretty wide spread of all these relatively, right? I don't have a measure to show you like this is 10 times greater. It's just a graphical representation. This one is about as far apart from this one as this one is from that one. Does that make sense? When they have a joining branch, like these 3, Pine, Horsehead, and Big Crooked, they join all right here on the same node. These 3 are more closely related to one another than these are, but they're not closely related to one another. Does that make sense? It's a relative measure. We do have a major distinction, which is listed here at the 70%. These east fish are 70% different from these west fish. showing significant stock differentiation in Wisconsin. This is telling you in our natural populations, we can expect different genetics. Each individual water has their own genetic stock. That's pretty cool. The real question is, what happens when you throw all kinds of other waters in there, especially ones that have been stocked? Does it turn into this mass of mutts, for lack of a better term? So moving on, they looked at all those 24 populations. Basically, it's the same graphic, except for I had to recreate these because the thesis was on paper. They didn't have an electronic copy for me. I had to recreate these as best I could from the paper copy. This is actually, I did it again, my voice is awesome. It's flipped, right? These are all the eastern waters on the bottom this time. These are western waters. The two that were influenced by stocking are listed here in green. They mis-ID'd where these go, right? These fish are more closely related to the western part of the state, Lake Tomahawk and North and South Twin. They are definitely not in that watershed. So we can see some introgression occurring in a couple waters of these very low stocked situations, but not in all of them, right? Those the 9 I showed you before were separate. If these were very close to being the same, they would be all in the same nodes. Yeah, that's another. That's like an okachi fish too. I think they're big fish there. I've never fished, need to maybe go. The next thesis and the last one, so bear with me here. The last one I want to talk about is Brandon Scudian. What he did is he took Ed Murphy's results and he added to them a whole bunch of other populations. So he added a whole bunch of 43 total populations. He doubled the sample size and just basically took any lake we were working on at the time. Get as many samples as you can. He wanted 50 individuals from each lake to best identify the genetic stock. So did the basically same analysis using a larger subset plus the stuff that Ed did. And what he found was that allelic diversity or stock differences existed across all populations and areas of the genome. 9 of the 43 had no documented stocking. That was in the Ed Murphy part that I showed. 4 of 43 showed significant impacts of stocking, but that doesn't mean that it influenced the population. Perfect example is the Coudray. That 1976 sample showed that it had an influence of stocking, but it didn't persist. The difference between these other ones, Tomahawk in particular, is that it does show an impact of that stocking. There is introgression going on there in Lake Tomahawk. There was also significant genetic differences between the sample populations. They were not panmictic, and what he found is we do not have one genetic across northern Wisconsin, at least in those populations, right? 43 is a pretty good sample, I guess, but when you consider we got 400 or 500 musky lakes up there, it's okay. Would we like to have all of them? Sure. Would we probably see more similar populations if we used all of them? Perhaps. But this is good news in that we don't have all one genetic makeup in northern Wisconsin. There was, again, a consistent east to west split. The outliers listed below, North and South Twin, Squirrel, and Lake Tomahawk, mostly because they're right there by the Art Emke hatchery, and got those type of genetics, but also got stockings from the Governor Thompson hatchery. So back in the day, what would happen is if we ever had surplus of muskies, there'd be an email that would go out. Hey, does anybody want extra muskies? And then I would type as fast as you could to say, yeah, bring them over by me. And this was before we had the genetic information, so we didn't know that it was probably a bad idea. And that's what happened back in the 90s. So that's why all these fish got spread all over the place, especially when you saw samples that went from Art Emke way over to the western part of the north. So we did find that there was a specific management unit that was recommended, the Upper Wisconsin upper chip, and then there was an inner chip that distinctly were separate from the upper chip fish, and that included moose, mud Callahan, and tiger cat fluids. Those looked really different compared to the other ones, and we're not quite sure why. We're still looking into some of that. But the overarching conclusion that he found was that because of the high degree of variation, most stocked fish do not contribute to natural reproduction in the populations. They didn't contribute to the gene pool. So these guys are visitors in these lakes. They get stocked, they survive, we catch them, they die. It's amazing. It's amazing to think that that's occurring in a lot of these systems. Now, it's not everywhere, but it's in the populations that they observe right here. So that's really good news. And now that we have more powerful information, we actually have set up a genetic management units with a genetic management plan so that we have a systematic way of stocking the lakes in northern Wisconsin to best conserve those specific stocks. In a boat. So, I can take some questions if you have them. I plan on sticking around. I definitely am going to have a drink. I also have, like I mentioned before, I think, I got a couple of the new baits that were from my friends at Lake X. I got the B52 here that just was released this week. I got another little bee that people want to look at. I got that TNA shredder that my wife beat up this year on Lake of the Woods. You guys want to look and talk about those a little bit. I also got some other little tips on some of the other dragons that what I like to do with them when I'm fishing them. I'm not necessarily here to talk about fishing, but I love talking about fishing, so I'm happy to do so if you want to do that. Yeah.
You want to comment on the poke in the gills theory?
Yeah, we talked about it earlier. If you saw, there was a post about, well, there's been a bunch of social media stuff about putting soda pop on bleeding gills of muskies and quote unquote saving their life. I think about these kind of things in this term. Let's try to use common sense, right? If you look at the pH of soda, depending on what kind you have, it's between 2 and 5. That's super acidic. Hydrochloric acid is 3. So soda is kind of like hydrochloric acid. So in my estimation, dumping soda on musky gills is like dumping acid on their gills. Or huffing hydrochloric acid into your lungs. I don't think any of us would last a real long time if we did that. And I don't think it's probably a very good thing for anyone to do to muskies. Now, there are some scientists that are looking at it. I'll be the first one to admit I'm wrong if that's the case. I would be very surprised if that's the case, just using physiology and the actual chemistry of the situation. So, my advice, if you get a muskie that's bleeding, do your best to let it go, but don't pour soda on its gills.
Another one was Gatorade.
I don't know what the pH of Gatorade is, but I imagine it's not.
Still said something about sugar.
pH on Gatorade's low.
Yeah, I would imagine, right?
It is.
Yeah,
Sugar with water or whatever.
you know, muskies have
Gatorade is so
blood cells just like we do that clot. So yeah, they'll bleed, but I mean, like I said, my advice would be just to get them in the water and hopefully they'll make it. We all have to realize that not every muskie we catch is going to live, right? We have to be able to live with that as muskie anglers. I've had one die on me. It was on Lake of the Woods. It wasn't quite legal, so I couldn't do anything. I had to watch the eagles eat it. It was sad, but I actually got some cool data from it because I have a contact there. I could call. I said, "Hey, do you want the clyster from this thing?" So the M&R had bones that could age the thing. So, but not everybody has that ability. But it sucked. 47-inch fish, go like a 55-gallon barrel dead. I didn't even hook it back. Didn't bleed at all. But it happens. But
Can you speak to fish that come up from deep water and have like the air bladder that like they try to go down and all of a sudden they come back up and they're just sitting there like they can't?
yeah, I mean, my advice on that is if you think you're fishing too deep, you probably are. Those fish are probably not going to survive. They can. What they can do is they will, unless they pop that air bladder, and please don't put a hole in it with a knife or don't fizz them, because that kills them. If it rips out of their inside, they're probably a goner. But if you would actually see their air bladder in their mouth, and they just can't seem to get down, just stay around them, give them time, don't touch them. And that's one of the things, on your panel, one of my biggest things that I've basically learned through time and through research is that, we all like the hero shot where we're leaning over the boat holding onto their tail. That potentially could have the ability to put more stress on the fish. And that's the last thing you want to do after you caught it. So if you're able, now I realize in some situations it's really wavy or it's really windy and the fish won't stay upright, but if the fish is going to stay upright, if you put it in the water and it stays upright, just don't touch it. Because they have, over their whole entire body, they have these tiny little cells that can sense. They can actually, they're so sensitive they can feel another fish swimming in the water. And when you have your hand around that caudal peduncle, they're like, I just went through this really stressful situation and now I can't move. And that's continuing to stress the fish when you don't have to. I got some flack. I was on a video on the Lake X page where I caught a muskie and I dumped it over the side like this, head first into the water. I got some hate mail for that.
St. Clair torpedo.
It's the St. Clair torpedo. They've been doing it on St. Clair for 100 years. Not 100 years, but a long, long time. You know, it's not having a significant impact on the mortality of those fish by doing that. It's better to get the fish in the water out of your hands than it is to hold onto the fish for an extended period of time.
Now they got revival teams on Sinclair.
That's a great thing. It's problematic with game wardens sometimes,
Especially here.
but most of our game wardens are pretty good. Like, look, I'm not trying to keep this thing. I'm trying to aerate it so it gets back in. The other thing,
Have their numbers that you just text them. and
Yep,
get.
we've got them on
Yeah.
speed dial here.
right. So
As club members.
I guess my point in this is, like, if you can, let go of the fish as soon as possible, because the more you handle it, the more stress you put on the fish. Stress kills more muskies than any 7/0 mustang is going to do. That's the easiest way to put it. The stress is a situation where stress is really bad for fish. I'll give you a perfect example. Our hatchery folks, they're different sort. And I've talked to so many of them that when they come in the morning, the first thing they do is look at the fish that they're raising. And if they're not eating, they freak out because they're stressed somehow. They either have a disease, the water's not right, something's happened, and the first thing a muskie does when it's stressed is quit eating. It's the first thing. And the stress can kill every one of those fish if they don't remedy the situation right away. So stress, it's not necessarily putting them in the net, which can have an impact. I actually just learned that recently. Different kinds of nets can have different impacts on muskies as far as mortality. There was a research study on bluegills that showed large knotted mesh was the hardest on bluegills. I would assume it's the same thing on a muskie. Fine knotless mesh was the best. Actually, no net was the best. To be real, right? But if we're gonna put them in a net, one with knotless mesh and small holes is generally the best for them. So all this stuff goes into it, right? And I realize it's all about, like, we don't all have unlimited resources. In fact, my net is large knotted mesh. I gotta buy a new net because I learned something.
But I look. Oh.
You wanna
See, and I
argue?
find, well, not argue, but there's also different things to it. Yeah, right? I mean, I've tried different nets too, and I found that some of the small hoop nets that supposedly are better for the fish itself or the slime or
They're a pain in the butt.
they'll grab your bucktail and they'll spin out, you know, now the fish is more stressed also because of the way that your lure and the fish react to getting it in the bag sometimes.
Yeah, it's about maximizing, and all I'm saying is, like, let's just try to be as educated as we can, and then we make our own decision based on what we can afford and what we can do, right? If a fish heads out of the water for 5 minutes because you're untangling them, that's not good either. But there's got to be some sort of, like, all I'm trying to do is just get as much information out there so people can try to make an educated decision. Any other questions? That was easy.
I got one.
No.
So, no, absolutely not.
Here, laser pointer.
Yeah, right. So you said that you can sterilize the eggs from the Fox River at the hatcheries. What's the potential for us? I mean, the broodstock lakes, Elkhart possibly coming up this year. We could also take the eggs from the Fox River down to Wild Rose. What's the potential for getting back up to that 25,000, 30,000 fish a year in the hatcheries?
We should get there this year with the eggs from the fox.
Up to 25 to 30,000.
Yeah,
Oh,
close,
awesome.
but they're not all necessarily going to go into green.
No, okay, but.
So
hey,
what
we're on
we're
the right
doing,
track.
so
Yeah.
what we're doing, and this is kind of on a different subject, but what we're doing is we're taking, for southern Wisconsin, we're taking all those gametes from the Madison chain. They're going to the hatchery, they're stocking the southern Wisconsin lakes. They're non-native lakes, those fish do fine down there, they grow big, we're good, right? We're taking all the upper Wisconsin fish, and upper chip fish that were stocked into the Great Lakes Basin and taking those ponds out of that production and put it into Great Lakes production. We're taking eggs from the Fox, put them in there, and we're moving towards stocking Great Lakes tributaries with Great Lakes gametes. Now, it's a slow process. Our intent is to do it this year. I'm going to talk tomorrow about it. Not a complete done deal, but we're moving that way. And what we would ask from a department standpoint, from clubs like yours, don't ask to stock fish that aren't that genetic in those systems. So, for example, a lot of clubs raise a lot of money to stock muskies, which is great. But if they're not stocking the right fish, that's a really tough position for us, right? Because we love the partnership that we have, and we don't want to say no. But if you say, hey, we want to get fish from this hatchery over here, and we know that this hatchery over here has 2 fish and spawns 2 fish that are garbage genetics, because we know that about some places, like, we don't want to sign that stocking receipt. And that makes you guys mad. We don't want to do that, but we don't want fish like that in the system either. We want the right fish in the right place. That's all we're trying to do. And while I can't always do everything that every angler wants to do, as a muskie angler, I'm trying to do my best to make muskie fishing as good as it possibly can be, and I got about 15 more years to do it. And then I'm going fishing.
Where are these little lakes getting their muskies from that are being stocked?
I guess I don't quite understand.
Just little no-name lakes, whatever, lake associations, where are they getting their fish?
Historically, well, previously, you know, they bought them from private growers. Gallons in Dodgeville grows muskies. A couple other places, not too many places in Wisconsin actually grows muskies to stock privately. But we've been approving these stocking permits. That is probably going to stop. However, what we do have in Wisconsin right now is we signed an aquaculture bill, which allows us to provide gametes to these private growers so that they can grow the fish that we want them to grow and sell them back to you guys. So we give them the right fish, they grow them up, they sell them to you. But they have to ask for them. So these private growers have to ask for the fish. Great question. There's language that says they have to pay for them, but it doesn't say how much. We can make it $1. And we don't, the point of the department, the muskie team, we're like, we just need to get these fish to these private growers so that we get the right fish out in the landscape. And then we got to make sure that the private growers actually grow those fish. Because it's not as easy to grow those fish as it is to grow the fish there.
Are you familiar with the CNR Club in Appleton? Yeah, I mean, so they take on, there's gotta be another John, I know is a member there, but they've been working on, is it Bass Lake? Boot Lake. So they've been stocking that one through their.
Boot and Vilas?
No, Boot in,
Forest County.
Forest. O'Connell Forest, it's,
Well, it's somewhere.
by Archibald, right next to Archibald.
Okay.
So, but they've been stocking that for 5 or 6 years.
5-year.
1 fish per acre, so it might be like 300 or 400 fish per acre.
Yeah, that's going to be problematic.
I was with a biologist though.
Well, I know that, but see, the biologist has been operating under a certain understanding, whereas this year is going to be the first year that, as you know, it should be a no, at least for the time being.
Or we should get the right stock.
Right, and so that's what we're asking the clubs to do is, hey, contact these growers that you're working with. Say, ask the DNR for the right fish.
Which
And then,
I assume
and then
Genetics
problem
is
solved.
a ceded territory too, correct?
Problem
Lake
solved.
is in the ceded territory?
I don't remember.
Well, if it's a stocked water, we would give them whatever proper genetic, right? It's in the upper Wisconsin probably, unless it drains to the Great Lakes, and then we.
It does not drain to get.
Okay, so then it's upper Wisconsin fish, and that'd be fairly easy for these growers to get. So regarding the broodstock lakes for Great Lakes fish, though, we need to, as a department, we're planning to get into all these to assess and see if we can get enough adults. The current thoughts are that we're not seeing the fish that we expected to see, but it could be, I mean, we could be wrong. So hopefully we get into all those lakes this year and next year and start using those in the fairly near future. Because those are all the St. Clair fish, right? Those are good ones.
Can you talk just briefly about your YouTube channel? He is also a content creator on YouTube. He's got a lot of great stuff there, not just musky catching, some good stuff there, but also some stuff from research that you've done years and years ago, some really cool stuff. So,
Yeah,
touch on that?
Thanks, Bob. I do have a YouTube channel. It's called Biological Angler Productions. It'd be awesome if you'd go and subscribe and like all that stuff. My intent, I have a lot of fishing videos on there. Like Bob mentioned, I have my surgery work that I did on the Manny chain where I actually put a radio transmitter in fish.
That's from when?
93? No, 2003. I was still pooping yellow at 93. No. 2003, I tracked fish on 36 muskies on the Manny chain. The summer of 2004 and the summer of 2005. Followed them around every week. Got to learn a lot about musky behavior. One of the coolest parts of that, and I plan to do a little bit more of this on my YouTube channel, is actually talk a little bit more about the science part of it. Just do really short clips of like, take a paper like I did before and summarize it kind of like I did here. Hey, this is what this research says. And then at the end, because this is what I do in my personal life, is I take the science and I say, okay, how can that help me catch a fish?
And that's what I was gonna get to next, is how can we take this information and incorporate that to our trips out on the water?
Perfect example of this is one of the largest fish I had a radio in on the Manny chain was a 46-inch female. She was 28 pounds. She lived, if you're familiar with the system, it's been long enough now, I'm fairly certain she's probably not around anymore. She lived in that small channel between Spider and Stone Lake. And nobody could catch her, including myself. And the reason they couldn't catch her was because that channel is inhabited by red horse about this big. And it was before all these giant plastic lures that we have now. I'm sure that fish just reached up, grabbed one of those red horse and burped for a week, or however long it took, and then went up and grabbed another one. That fish, though, the part about fishing that it taught me was one spring, I was in Stone Lake following her, and I got close enough with the outboard. I had a 25-horse Johnson on a 14-foot Alumacraft at the time. I motored up to her. It was kind of behind the island, just real stumpy back there. And I was going, I'm like, oh, I better put the trolling motor down. And I kept following that fish and kept following that fish, and I couldn't catch it. And it was going along the shore, and I'm trolling motor on 10, couldn't catch it. Finally, like, whatever. Pulled up the trolling motor, started the big motor. Motor ahead 100 yards, she's laying right there. She was running away from my trolling motor. They also don't like electronics. I'm not going to say that they have a brain because it's this big and their fear, fight or flight type response, but they definitely understand bad experiences and they react to certain stimuli in certain ways. The other thing that I learned about those fish is about half the fish I had radios in out there were open water beasts. They were always in open water, and they were in all different lakes in the chain in different parts. And if you were out there fishing for them with your electronics on, and specifically multiple electronics going at the same time, you were much less likely to interact with those fish than if you didn't have any electronics on. Now, I love electronics. If my depth finder doesn't work and I'm out there, I go home. Except for when I'm open water fishing on the Manny chain, or open water anywhere. Shut them off. You don't need them. They like the black crows.
Okay.
A little different than Wu-Tang.
I bet you they like Wu-Tang. I like Wu-Tang. Rage, they like Rage. So, yeah, I mean, there's probably 1,000 things that are up here about that research that I could tell you about if we talk long enough. Being able to follow 36 muskies for 2 summers, it's invaluable. Very few people get to do that. So.
And that type of research, that's radio telemetry stuff that was done decades ago, similar to what we're doing now on the Bay.
It's going to be super telling.
Yeah, with UW Stevens Point. So I suggest you go take a look at his YouTube channel. Like I said, a lot of great stuff on there. Also shows you the behind-the-scenes stuff that we don't necessarily see.
It's not associated with the DNR. Sorry, I got to say that.
That was for your your master's or undergrad?
Well, that was through UW Stevens Point, but like my YouTube channel, what I say on there doesn't represent the Department of Natural Resources.
Personal channel.
So I have to say that. Sorry to interrupt you.
No, not a problem.
I would have forgotten.
I guess I gotta ask, Shauna Lake, I know they're not putting in the Great Lakes strain, obviously, should they be?
They will be.
They will be, going forward? Okay.
When I say they will be, I'm not guaranteeing you that it's gonna be this year.
Okay.
What I would ask in this situation is to be patient, because the demand for the number of fish far outweighs what we can produce. And we typically stock muskies on a biannual rotation. So, we may stock Shawano next year, and then it would be 2 years before it gets fish again. But that's typical for all our stocking, minus Green Bay. Green Bay gets fish every year. Green Bay is our primary responsibility. It's our primary reason for Great Lakes fish. It's our number one priority. Green Bay is the reclamation project that we're most important part of our Great Lakes program. If we can raise enough fish to stock these other waters too, we're gonna do it, but we're not also gonna stock all of our fish in Green Bay, because it's just not.
At any point, are we able to help with the Green Bay fish as far as like bait, or to, get them another week or 2 weeks worth of bait, or raise funds or something like that? I know
Aren't you guys already doing that?
the Alliance helps with a lot of that stuff too.
Absolutely. I mean, the Musky team, as the leader of the Musky team, I set up a gift account that we can accept checks and donations from various clubs for that purpose. The problem we run into with that situation is most people want to say, and this happens more in the north, they're like, hey, we want to stock XYZ lake, can we get the hatchery money to raise our fish? And I can say, well, you can give us money, but I can't guarantee they're going to be your fish. Like, because a pond raised 5,000 fish. We don't stock 5,000 fish in anything. So we would be feeding all the fish. It's kind of like a greater good thing. If you're willing to donate, great. We'll say we guarantee they go to this stock of fish, so it would go to the appropriate hatchery. They would be fed to those fish. It's for the greater good. They would be raised. But yeah, I can accept those checks. The hatchery folks can do it. I can tell which hatchery what to use and how much. And so, the other cool thing that I've been doing is working with pit tags and folks. If you guys are interested in getting pit tag readers that are really fairly inexpensive, did we talk about this?
We did that in the Alliance.
Yeah, there's a pit tag reader from Sweden.
We purchased a
That's
bunch
where
of
we
those
got it.
last
Okay,
year.
excellent. So you already know about that. My boy Marcus.
Yeah, that information was brought back to
Okay,
the club.
perfect.
So thank you.
If you have problems with those readers, I've had probably 40 that I've purchased. I've had one that just eats batteries like the Oreo cookies. I called him. He's like, next time I send stuff, I'll send you another reader. So he's super good about it. If you have any that malfunction, let me know, or let Marcus know. He's super good about it.
Anybody here
how
interested in
much are
pit
they?
tag readers for 2020?
They're about 150 to 170 bucks versus 600 from a U.S. source.
Yep. The club actually purchased some, and you can sign them out basically and just bring them back. But I know a couple guys have also purchased their own for their personal use.
I just want to thank you guys all. I know it's been long. I always talk too long. I'm sorry, but oh, I'm all right. I'm thirsty.
One last item. I just want to make sure that everybody understands here that Jordan is also going to be participating in our panel discussion, catch and release A to Z at the Wausau Expo. So anything to do with he's going to be covering a lot of the stuff that we talked about here today. We're going to be covering a lot of biology, physiology, what's best handling methods. So please stop by, check out the expo, check out the seminar, stop by our booth. The more the merrier. And I just want to also thank Jordan one more time.
Thank you guys. Thank you very much. I got cards up here.